Phenotypic Heterogeneity of Genomic Disorders and Rare Copy Numbers Variants. Some copy numbers variants are associated with genomic disorders with extreme phenotypic heterogeneity. The cause of this variation is unknown, which presents challenges in genetic diagnosis, counseling, and management. Now, let's going to see a video about this.
Hi, my name is Ryan Benowitz. My project is developing a learning classifier system for the detection, modeling, and characterization of genetic heterogeneity. Genetic heterogeneity is when you have different subsets of people within a population that have different underlying factors which cause the same kind of disease. In our lab, we're most interested in pursuing autism as it associates with genetic heterogeneity. And you can imagine a set of autistic patients where some group of the autistic patients have a specific set of underlying genetic causes, while an entirely different set of autistic patients have an entirely different set of genes and etiology. A few different methods have been attempted to approach genetic heterogeneity, including clustering and stratification, which, while effective in dividing up your population into smaller groups, is not guaranteed success in capturing that underlying heterogeneity. And in fact, when you reduce your population into smaller groups, you are now looking for a model within smaller sets of patients, and that reduces your overall power to find a good model within each smaller group. And so in our lab, we are interested in identifying the genetic causes and also environmental causes of common complex human diseases. The value of using the computational power of a massive number of processors in, let's say, a supercomputer is that it allows us to approach this extraordinarily complex problem. It's very important to develop algorithms and to advance technologies to allow faster and better computation. Specifically, the algorithm that I work with, that I'm developing, is something called a learning classifier system. And the idea with a learning classifier system is that you develop a whole population of rules that represents your solution. The normal paradigm in genetic epidemiology is to identify a single best model to explain whatever phenomenon you're trying to explain. And with learning classifier systems, they evolve an entire set of interacting rules which collectively make decisions, as opposed to a single rule that makes decisions. We think that these learning classifier systems lend themselves very well to our problem, being that we have a problem that has multiple solutions. So with that in mind, we want an algorithm that allows for multiple solutions to evolve. And thankfully, with the funding of the Newcomb Institute supporting this research, we hope to identify the specific underlying etiologies and identify distinct subtypes of autism and uncover the specific factors that play a role in those smaller subgroups. The exciting part about this research is that it should be applicable to not only autism, but any other common complex disease where genetic heterogeneity occurs.
Chromosomes can vary in a lot of different ways across people and within people, and everyone knows about certain types of copy number variants. For example, people with Down syndrome have 3 chromosome 21s. That's one very big copy number variant, but the chromosome is made up of repeats of different structures within the chromosome and even deletions of parts of the chromosome, and there are all sorts of diseases in pediatrics typically where you could be missing a little piece of the chromosome. And there's also very small copy number variants, and then even smaller than that, what's called sequencing of genes, where you just find one base pair. Now that may be a lot of concepts because you'd need to know a lot about the structure of genetics, so this may be far more in depth than I think you want to go. But the kind of copy number variants that we've been studying, which are just changes in material in the chromosome that are too small to see with a microscope, but can be seen in new ways that have been developed to scan all of your genes, the entire chromosome. And we're not just looking for copy number changes in parts of the chromosome, but we're looking for parts that are very rare, because there's a lot of normal variation, just like people are varied in terms of how tall they are, what color their eyes are. There's a lot of healthy variation between people in terms of these very same variants. So when we find a copy number variant in, say, a group of patients, as we have and are still doing, we first have to look it up on tables to make sure it hasn't been reported before in normal subjects. And this is such a new field that the database for the normal variation is growing all the time. So this is a work in progress, because we'll find something that we think is rare, and then over the next year it's found more than we thought it would be in normal populations.
Genomic rearrangements are an important source of genetic and phenotypic variation. Rare, recurring copy number variants of pathogenic significance. Term genetic disorders were originally identified in persons with a characteristic set of clinically recognizable features such as the Smith-Magenis syndrome, the Soto syndromes, and Whelan-Barnes syndrome.
I don't think it is necessarily. There are certain parts of the chromosome that seem to have this more than others, and we're only been particularly interested in what's called the coding regions, the part that make proteins, and people have tended to look at this selectively. Many of the variants that we're finding in that science paper where Walsh was the first author, 80% of the ones we found were inherited from healthy parents, and that's generally not reported in the other studies because they don't necessarily have trios. But when you have trios, meaning you have a mother, a father, and a child, you're finding that many of these, in fact, are turning out to be inherited, which means they may still be important, but they're probably, for many of these, a risk factor, not necessarily the cause of the disorder.